as a special guest from a group. Adriana has studied in Brazil, in the Federal University of Rio Grande do Sul. She has studied agriculture and environmental microbiology and has done PhD in genetics and molecular biology there, and then moved to Florida to do a postdoc for 3 years, working on microbial ecology. And then she moved back for a few more years to Brazil, and now she's spread in 2 institutes in Germany. The Leibniz Institute of Vegetable and Ornamental Crops in Großbeeren. I think I know the place.
You know?
Yeah.
Far away.
It's in the middle of nowhere. Yeah. And then in Braunschweig in Germany. But you're moving to Berlin, actually. You're on the process of moving to Berlin. Yeah. So Adriana worked for a couple of years with Cornelia Smalla from Braunschweig, and she has a broad focus on microbial ecology, microbial communities with respect to soil and plants. And we are so happy to have you around and looking forward to your talk. Thank you very much.
So thank you very much, Jan, for the invitation, and also Caroline, because I know her for almost 10 years already. I was a co-advisor of her in Brazil. So that's why the connection was possible. And then now we want to spread a little bit and go into different directions, but all are focused in microbial communities promoting agroecosystem functioning, soil and plant health. You guys, I think most of them, you don't know me. So I'm from Brazil, and I'm a microbiologist, but I was forced to do bioinformatics, I think 20 years ago. It was totally against my will, but I think there are things that come for good, and I get a lot of profit from this. So I don't consider myself a bioinformatician. I still want to be known by a biologist, microbiologist, but I think it's a big part of my projects, or I mean, all the projects are related to understanding the microbiome through bioinformatic tools. So just I doubt you know this place, because, you know, it's in the middle of nowhere. When I got the job there, they told me, okay, it's near Berlin. Actually, it is, but it takes 2 hours to go there. So it's in the middle of nowhere. It was a research station during the DDR. So it's more than 100 years a research station, agronomic research station. It's very close to Berlin. It's like 30 kilometers, but it's very hard to reach the place. And the focus there is resource-efficient agri-food systems, health, nutrition, and food security, biodiversity, and pesticide reduction, and the climate change. Those are our goals there in our research. And then we have these 4 groups, big groups: ADAPT, that it's plant adaptation, biotic with biotic interactions, quality is food and plant quality, and the horti-systems, because we are one of the only horticulture institutes in Germany. And also we have the science support platform. I just put it here. If you have any interest, you can look at the website. But we have this science support platform that works with proteomics, metabolomics, with LC and also GC. And they are doing a lot of things in plants and microbial communities there. So in 2017, FAO wrote a report saying that we are going to face a lot in feeding more than 10 billion people that is expecting by 2050. So I just highlighted here for my research, and I want to share with you the first 3 challenges that they highlighted in this report. So we need to have a sustainable, improved agriculture productivity to increase the demand. We need to ensure a sustainable natural resource base. We need to protect the environment. And also we need to address climate change, because it's not something that we need to deal in the future. It's already here. And we need to take this in consideration in our research. So then we have those challenges. besides all the other challenges that I can highlight: climate change, soil erosion, biodiversity losses, antibiotic resistance, plant diseases, and the use of pesticides. So we can, in our research, and I try to summarize what I have done for the last 10 years more, but let's pretend I'm very young. So the last 10 years, so I have worked with organic farming and conventional farming. So what is the microbiome dealing with this kind of situation? Ambient and future scenarios. So as I said, the climate change is already here. We have some ways to measure or try to assess the impact of this climate change. And also crop rotations and monoculture. I don't know here in Czech Republic, but in Germany now there is the rule that you cannot have monoculture in crops anymore. And so this is something that needs to change. It's a transformative change in the society that you cannot deal with this system anymore. It's not the best for the issues that I have mentioned before. So to have a sustainable plant production, we need all of this. Beneficial microbes, and here includes the ones that I work. I'm a bacteriologist, so I work with plant growth promoting bacteria, but also the AMFs, because my host here works with AMFs, and those are also important in our research. So we study the plant and soil microbiomes. The suppressiveness of soil, it means the ability of the soil to suppress diseases. This is also very important. Control of pathogens. Dysbiosis, that is a term that is very important in human microbiome because of this disbalance in the gut, and causes many diseases. And now they are relating this dysbiosis to even brain or neurological diseases. So we also have this in our research. And climate resilience. And we know that the global climate change is already here. Yeah, and it's not only the polar bears that are suffering from this. You know, so our agrosystem with the microbes, they are also suffering from this. So we have the direct and indirect impacts. But the extreme weather, they have an extreme impact in agriculture. And then we have to face, as FAO is already highlighting in 2017, that we are going to deal with decrease of grazing areas, decrease of soil fertility and the availability of water. We are going to change the way we use the lands, and also ecological changes, because as I said, it's not only polar bears or the pandas that are suffering from this. We have the microbes, we have the humans, and also the increase of diseases, especially fungal diseases. There are many papers that highlight the increase of diseases, mainly fungal diseases, is going to increase of the climate change, the way we are facing this now. So how do the plant and soil microbiome deal with plant pathogens now and in the future climate scenarios? Here are the ones that it's just a few of the papers that says that we are going to have more pathogens, more plant pathogens with this climate change. We're going to have more humidity, although we have more drought also. So it's going to be a mess, if you can say. And then we also are interested in understanding, I don't know where to point at this. Okay, it's not where I think. The dysbiosis, it's going to have a big impact. And the dysbiosis is what goes against the postulate of Koch that said that for each disease, there is an agent. In this dysbiosis, it's not what we see. We see opportunistic microorganisms or good guys that were in a bad situation, or the climate and the situation is not good enough, and then they causes dysbiosis, and then disturbance in the microbiome, and then they causes disease, or they made a door for new fungi to arrive. So how do we assess this, all this microbiome in this changing environment? So we need fields or greenhouse experiments or growing chambers, because we need the plants, right? So then we have experimental design, sample procedure, and we have, of course, a good statistics. We have the plant itself, and the plant, it depends. You can have crops, you can have, in the case of the IGZ, or IGZ, you can have mainly vegetables, that is the main goal there, or ornamental crops. And then we can measure disease severity rating, pathogen quantification using qPCR. We measure plant parameters like shoots, fresh weight, dry weight, nodulation rates, and then root and fresh dry weight for the plant. So we have the fields, we have the plants that we want, and of course we have what I like the most, that is the microorganisms. And in the microorganisms, we have the microbiome, and I don't know how to highlight, okay, let's see, here. So we have the microhabitat sampling, so we can select the part of the plant that we're going to assess the microbiome, because as humans also we have the microbiome is different from each part of our body. Yeah? And then we have the taxonomic and microbial ecology and the amplicon sequencing that we use to assess the microbiome. And there, let's see, here, we have the bioinformatics that can assess the microbiome from the samples. Then we also, and it's very important, and it's not, we cannot exclude this, is the bacterial cultivation or the microbial cultivation in general, because we need these bugs in our plates. Yeah, we need them to understand. So it's not because we are using something sophisticated as amplicon or shotgun sequencing that we are not going to assess them in a traditional way. So we need cultivation. We can do culturomics. We can do, or conventional, that is one by one, culturomics that we take a bunch at a time. We can do comparative genomics and also metagenomics and whole genome sequencing to understand the genome of these bugs. And then we go to a different direction that is transcriptomics and things like that, but this is something that comes later. And also, we use these microorganisms that we are isolating because we need them. And then we have the synthetic communities. The last conference I attended, Microbe in Vienna, they suggest, a person suggested that we use simplified communities instead of synthetic communities, because synthetic looks something not nice. That's what she said. So then I still use synthetic community because it's what we are more used to, but simplified communities are also a terminology that is raising up. And then that's what I said that I didn't like, but then I like, and then I get used to. So I do a lot of analysis in silico, and I use Dada2 to analyze the amplicon, because it's the state of the art of the amplicon sequencing, especially when we have thousands and thousands of sequences at the same time. We are I always fight for open sources and things that can be done. Yeah, in any case, I come from a country, a development country, so I always have in mind that it cannot be just for elites. We need to have science for all. So we try to accomplish this. And we use Silva, Unite, and when we are dealing with protists, the PR2 database to assess the microorganisms. And then I work with mainly 3 soil-borne fungi: Fusarium, Genamomyces, and Rhizoctonia solani, because they are the most important, not Rhizoctonia, but these 2 are the most important diseases in wheat. And since I am in Germany, that wheat and barley is very, very important there. So those are the 2 main pathogens that we worked. But Rhizoctonia solani is very important for other crops, and then we also always added to the experimental design. And what I told you about the microhabitats, because you know what you have in your gut is different from what you have in your skin, and it's different from what you have in your head. So then it's the same for the plants. Yeah. So there is a gradient of microorganisms. So we have more, the diversity is higher in the soil, and then it goes to rhizosphere, and then the plant select. So there is a selection from the plant to define who's going to be very close to the plant, and then deal and communicate using the exudates of the plant and also from the microorganisms. So basically in our research, we do bulk soil as well, here. because we also want to understand what's going on without the plant, more or less like a control. And then we have rhizoplane, that are the bacteria really attached to the roots, root-affected soil, that is around the roots, but not on the roots, and the rhizosphere, that is the most famous microhabitat or compartment, that is the ones that are nearby the plant, but not really, really attached. And then we have many projects, missing here. So we have many projects, I'm not going into detail, but those are the projects that I worked during my agriculture life. So microbiota in healthy plants, plant microbiota in abiotic, and plant microbiota in biotic stresses. And then I highlighted some plants, and there I put the black in that were finished. The blue ones are ongoing, so require still some data processing and writing papers. And also the red ones are the ones that I have just submitted for grant agencies. So in Germany, they have what they call Eiweißpflanzenstrategie. That is, they want to focus in legumes, and they want to focus in protein crop. That's what it means, protein crop strategy. So they want to focus in pulses, because legumes can be divided in three tribes: the green ones, the pulses, and the oil, that is soybean and peanut. And then they want to go into Germany is already the legumes. I think it's peas and faba bean is the most important legumes there, but they want also for human consumption. Peas is, but not faba bean. And then they want to bring some opportunities from the cultivation, so we are into this, and also environmental protection, so we need to protect the environment, and also they also are in this process of, I think, the European Union that wants 25% of the land to be used as organic farming. And then we have, I highlighted in the other slide, that is, we have this project that I just submitted yesterday. So it's beans for DIV, that is for diversity. It's in the Biodiversa call. And it with common beans from South America and also from Europe. Luneta is white lupin. I don't know if you have this lupin here, if you have cultivated, but it's already a big deal in Germany, and it's very important because it's very nutritious in terms of protein, and it looks like, I mean, in terms of protein content, it's like soybean without the oil. And also, I'm writing a DFG, that is also our Germany agency, for pulses. And then I want to take pulses with different life cycles, the perennials, the annual and biannual, but it's still in process. And then beans for DIV, or for DIV. This, I'm not going into detail because I talk a lot, but I know that the clock runs. This is the experimental design that we just submitted. So we have different varieties. And in this case, we are interested in land races, because we know that the land races have a special microbiome in other crops also. It's in the literature already that when the crops went into breeding process, the microbiome was not taken in consideration at that time, because it was 100 years ago, 200 years ago. They didn't take this in consideration, and still they don't have the microbiome as a big impact in the breeding calculations that they have. So we are trying to get the land races back, but not to convince the farmers to use land races to substitute the high yield beans that they have, but as a crop that can restore the microbiome, especially in a regenerative agriculture. That is something very interesting and hard to implement, but I think maybe it's the future when you can use small farmers like in common beans, not big monocultures. In Luneta, we are adding the looping to the cropping system. So we have a cropping system where we have brassica that has some fumigation activities, and we have we have lupin that provides nitrogen, and we have wheat that is the main crop. And then also everything is related to assess the microbiomes and also try to, in this case, we have a synthetic community that we can use and provide a source of inoculation for. So this project is being evaluated now. So we got already the second phase, and let's have the fingers crossed. And we have something very nice that is not related, it's very hard to implement in Germany, that is coffee. So we are in a project with the Brazilian collaborators, because this is the good part of being an abroad scientist, you have connections there. So we are in the metaomics of coffee canephora from Amazonia. We are used to coffee Arabica, that is the most produced in the world, but coffee canephora, or you might know as coffee robusta, it's a very important because it can deal with higher temperatures. It doesn't require mountains to grow as the coffee Arabica. And this is great for climate change because climate change is already here. We need to find alternatives. So then they are using old farms. They promise us that the Amazon is not being destroyed to plant coffee, because it's the first thing you think. If they are taking the trees out to have coffee, we don't want to join. No. And then they have statistics and they have good research there saying that it's from old farms and old grazing areas. So we have the coffee canephora, but although coffee is not native from Brazil, it's hard to bring soil to Europe to do any kind of analysis. And then in that case of the project, we are assessing the metabolomic and the microbiome from the cherry beans. That is the first cherry after when they put it from to dry. So we are assessing because of the terroir. That is the very local, what the local microbiome or local things from the soil can contribute to aroma and flavor. So we are going to have different fermentation processes there, and then we had already, and they sent us the cherry beans, and then we are going to make metabolomics, and then we are going to assess the substances and also the microbiome from this. I just shortly want to show you the two projects that I just finalized. They ran together in Germany. It's Super Soil. It's a biodiversa also. Professor Cornelis Mahler was the coordinator from the German part. And also Rhizoweed, she's also the coordinator. But both projects just finished in the beginning of the year. One is the suppressiveness of soil against Fusarium mainly, but also GGT, and the rhizoweed is wheat, and the rhizosphere processes and yield decline, because we know that in wheat, after some monoculture, the yield decrease, and they want to understand why. So we have 3 different locations in Germany, and we have this is okay, and this is another thing. This is another finalized project that we have, and that's why brings Caroline to this, and also Sydney Strumer, that is from Blumenau. And we have 2 papers with them already, because as I said, I was the co-advisor of Caroline. And then now we just submitted the last paper of her thesis. So I hopefully it's going to be processed soon. And where we assess we have inoculation with AMFs, and then we assess the microbiome. So I think it's a very nice, it's a little bit a part of agriculture itself, but it's regenerative soils. So we are also interested in recover soils, and then we can have healthy soils and healthy plants and healthy humans, and then we go into this One Health system. But the super soil I don't know if you know this place, but it's the Global Change Experimental Facility in Halle. This is what I called a time machine, because it can simulate the climate change that is going to happen in Germany in 80 years, more or less. So they have computational modeling scenarios, what is going to happen in Germany, and they increase the temperature in 1 or 2 Celsius, and also they change the precipitation pattern there. So we have more in spring and fall and less in summer. And they have this very nice greenhouses that are not, it's just roofs. They open and close depending on the weather. And then they simulate what they expect in terms of raining. There, they have plots for ambiental and future scenarios, and conventional and organic. And then that's what we sampled. So those are the samples that we brought the soil to the greenhouse. And then we have a lot of greenhouse experiments with this soil. And then we used wheat, because it's our lady for the projects, Fusarium and GGT, that is our Gammamomyces graminis. And then we did a lot of bacterial isolation, because, as I said, it's important also, it's not only amplicon sequencing. So we tested, a lot of them were positive for phosphorus solubilization. They produce siderophores, they release siderophores. They also, in conventional soils, they are more isolates positive for tested enzymes, so it seems they are more active. And we have many pseudomonas in the organic farming soils that are promising also to go further in these studies. So the conventional farming soils with GGT, there is higher disease in conventional compared to in future scenarios. In conventional farming, the diversity in future climate scenarios is higher than in ambient climate scenarios. And the treatment with pathogenic fungi decreased the soil microbial diversity. So those are preliminary data. We still have a lot to do on this, but we can already work with these ideas of what is going on in the future. In the organic, the diversity in future climate scenario is similar to the ambient climate scenario, and treatments with pathogenic fungi decrease the microbial diversity. So it seems that conventional and organic, they behave different, and it's in general, it's going to be, we can advocate for organic farming, because it seems that it buffers that somehow the impact of the climate change. When we look at the composition in conventional farming, the composition of the microbial diversity in ambient and future is different. And the treatment with pathogenic fungi modifies also the microbial composition. And when we look at the organic farming, the composition of the microbial diversity in ambient and future is more alike, but the treatment with pathogenic fungi modifies the microbial composition in different ways. So we might have seen here something that is close to the dysbiosis, because I didn't mention, I skipped this part, but there is a concept that is the Anna Karenina principle, because it's written in the first page of the Tolstoy book, Anna Karenina, that most happy families are alike, but the unhappy families are unhappy in their own way. So there is a Anna Karenina principle that says that it can be used for microbiomes as well. So there are in humans first, and now in plants. So the stressors, they behave different depending on what you have. So climate scenarios, organic farming soils might be less affected by global climate change. Phytopathogenic fungi impact the wheat microbiome in both farming managements. And then here we have to speculate the dysbiosis that is going on, because we cannot distinguish any kind of pathogen that pop up. And further investigations are still required to measure the impact of this in future and climate scenarios. So we're going to keep using the experiment station there. For rhizo wheat, it's a different question. We want to understand how is this spatial effect how depth can impact the microbiome, and also if the yield decline has something to do with the microbiomes. So we have a paper in Frontiers about that. I'm not going to get into detail, but it's wheat rotations, because we are, as I said, the monoculture decreases the yield. So we have short rotations that they rotate with rape seeds, but also we have, and in this case, we have a core, a soil drilled that have like 120 centimeters. And then we can slice as we want, and have the, so we had in this case, we have 5 soil depths. We have 3 microhabitats, the ones that I mentioned, rhizoplane, rhizosphere, and root affected soil, and we have 8 cores. And then, yes, the microbial composition varied in composition and diversity depending on the soil depths and microhabitats. And we could also name the microorganisms that were different. It's not a big deal, but it's important to describe and try to understand more about the microbial community. So the most abundant taxa belongs to the proteobacteria, actinobacteria, and also many, many potential plant growth promoting species. Now what we want to do is to try to understand if these microbes came from the surface and go down, or if they are being recruited from the surroundings, from the below. So this is still something that we need to evaluate. And then they might be essential to cope with biotic and abiotic stresses, even in deeper soils. Because, you know, if you have a dry soil, you have more roots to go and try to get water. And then the microorganisms there that are in this portion, they are very important. The other question in the rhizo wheat was if we have beneficial microbes that accumulate in wheat monocultures, if they can deal with the pathogen that we are doing, because the yield decline is caused by general shift in microbial communities. So we also publish now, it's in the phytobiomes, but I didn't know, but it takes more than 6 months to get a preprint or something like that. So now we have just what comes from this short information but at least we have the DOI already. The microbial community is varied in diversity in composition and diversity depending on the site and microhabitats. So again, we have the microhabitats, we have the different in 2 different soils, silty and sandy loam in this case. So we have soil plant continuum, that is this from bulk soil to rhizosphere, from root affected soil to rhizosphere to rhizoplane. So we have differences in this, and rhizoplane harbors very specific taxa that is expected because the plant is selecting who's going to be close by. We had more than 700 isolates from different microhabitats, and they have a lot of activity that are interesting as plant growth promoting bacteria. We are interested in particular in Actinobacteriota because it's a very important microorganism in soil. And we have them tested at the greenhouse, and they indeed help mitigating the impact of the fungi at the greenhouse. So we use the isolates that we, as a synthetic community, and then we are very happy about them. So continuous wheat, that is the monoculture, changes microbial structure. It doesn't matter if it's sandy or loamy soil, and also sampling years, because I forgot to mention, we have a year with drought and a year without. And we also assessed the soil plant continuum, that is this selection of the plant. And continuous wheat accumulates the take-all disease, that accumulates the fungi in the soil. So it's also something that advocates against the monoculture. So here's just a summary of the climate scenarios. Organic farming soils might be less affected by global climate change. So this is something that we can also use to change the mind of the politics at the end. If we have to deal some kind of dysbiosis, this is something that we still need to assess. And it's very hard to assess the dysbiosis because it's not, as I said, it's not one guy doing something. No? And but we still need to investigate more. And there, yes, there is a lot of PGPRs in the rhizosphere of wheat, and even at 120 centimeters. So it seems that they are there following the plant, and it can be a good strategy to cope with nutrient and water limitations in the climate scenarios. So here I increase a little bit my poster. Now I have also these Mesoamerican and European common beans. That is going to be this next project. That is the beans for DIV, because we know that they have different species. It's not. I think it's not called a speciation event because it's not turning into a different species. It's also Phaseolus vulgaris, but we know that beans that they come from from Mesoamerica, they have the Andes, from the high altitude beans and the low altitude beans. And then we have the ones that came here in Europe 100 or 200 years ago, or maybe more, and those are following a different genetic way. And now we are getting the land races from these two, as I mentioned, to try to retrieve the biodiversity from the soils. And also the annuals and perennials, that is something that I want also to go into detail. And then again, we have all of these things that goes in our, so we always try to keep this, I have this always, I have it printed in my wall, to keep in mind that we have to keep this as part of the research. Climate change is already here. It's not something that is going to happen soon. No, it's there. And we have to implement this in our greenhouse. Because this guy said, Albert Howard, the health of soil, plant, animals, and man is one and indivisible. And then why the One Health is so important is I think is the top words from the last years, because we cannot separate anymore. It's an illusion that we try to solve one problem and not the other problems. So those are the groups from the different institutes. Below is the IGZ, more or less. It's a little bit mixed. And the guys from the JKI, where I had for 4 years, and the funding agencies. And I thank you very much for your attention. I know that is not the time for the seminar. You have another hour of sitting and listening, but I appreciate a lot that you have time to come here and see me. Thank you very much.